everyone. We will go ahead and get started. Just want to welcome everyone to the AOS Student Research Grant Seminar Series here today. My name is Robert Driver. I'm a postdoc at Duke University, and I'll just introduce the seminar series first here. So, each year, as you probably already know, the AOS offers funding to students and early career researchers with the AOS Student Research Grant. And the AOS Student Research Grant Seminar Series, where we are today, celebrates the challenging and innovative work of the AOS Student Research Grant recipients by sharing this AOS-funded work to AOS members and the public. And each week, we showcase the research and research plans of a 2024, 2023, or 2022 AOS Student Research Grant winner in a seminar-style format. And we are almost midway through going each week until December 15th. So I want to thank everyone for being here. Thank the AOS and the AOS Student Affairs Committee and Chris Ann Handel and Ramona Fletcher for their support and input, and especially thank Megan, our speaker today. And before we turn our attention to Megan's research, I just wanted to mention if you have questions, there will be time for Q&A session at the end of Megan's talk, and you can participate by unmuting yourself, by raising your hand icon, and then I can unmute you to ask the question, or you can type the question anytime in the chat box, or there's also a Q&A box to type in, and I'll monitor all these three methods of asking. And I'd like to remind everyone that this is an AOS sponsored event and that all participants are required to follow the AOS virtual meeting event code of professional conduct. And I will post a link to this code of conduct in the chat in a moment. And I will also post a link to a post-seminar evaluation form that you can fill out. And this really helps us improve our seminar from week to week for you to add suggestions. So I'll post that link, and then also a contact email for the AOS Student Affairs Committee for any questions. And please be aware that we're recording this talk. Okay, so today, Megan Miller is a Master's of Science student in biology from Colorado State University-Pueblo. And Megan's academic career began with a Bachelor's of Science in Fish Conservation and Wildlife Biology from Colorado State University in Fort Collins. And following her undergraduate degree, Megan worked at a number of avian technician field jobs all over the United States, including in Massachusetts, New Hampshire, Virginia, Colorado, Utah, and North Dakota. So wide variety of habitats and species, I'm sure. And during this time, Megan banded and oversaw. So I had to do a calculation here. During this time, Megan can correct me if I'm wrong, but during this time, Megan banded or oversaw the banding of over 4,500 individual birds being banded. So really incredible work. And portions of her research are published in Avian Conservation and Ecology. And in 2023, Megan returned to school as a master's student in the lab of Dr. Claire Ramos at Colorado State University-Pueblo, where she is now. And in addition to the AOS Student Research Award, Megan also won the Paul A. Stewart Grant from the Wilson Ornithological Society and the Bergstrom Grant from the Association of Field Ornithologists. And this is all in the same year, 2024. So great work. And Megan is currently a banding technician at Rocky Point Bird Observatory in Victoria, British Columbia, while she's finishing her master's degree. And so with that, I will turn it over to Megan for her talk entitled Modeling the Effects of Wildlife Smoke, Body Condition, and Diet Guild on the Oxidative Status of Migrating Birds.
All right, thank you so much for that introduction, and thank you everyone for coming, and thank you for that mouthful of a title. You did great. I will shorten it at some point. So obviously I am Megan. I am a student at Colorado State University-Pueblo in the lab of Dr. Claire Ramos, and today I'll be talking about the effects of wildfire smoke on the oxidative status of migrating birds. So I'm going to start kind of big here and work much, much smaller. So we're going to start on kind of a global scale talking about wildfire smoke and how changing fire regimes are increasing our exposure to wildfire smoke. So these changing fire regimes are extending the fire season later into the winter, especially here in Western United States. It's changing the fire frequency and duration in different habitats. So habitats that are accustomed to seeing fire are seeing less fire, and habitats that are not accustomed to seeing fire are seeing more fire. And this is increasing human smoke exposure in a variety of places. I think we're all familiar with the severe pollution that occurs in places like Los Angeles, and even Colorado and Denver has some pretty severe smoke days in these like major fall wildfire events. And then Australia and the Amazon are also experiencing these kind of major wildfires that produce these enormous smoke plumes. And there's already a lot of medical research being done showing that this wildfire smoke is impacting human health. And especially during 2020, it was shown to kind of exacerbate the conditions of COVID and potentially increased the number of deaths because of the combination of COVID and wildfire smoke. So humans, for the most part, are very lucky that we can take shelter in our homes. We can wear a mask. We have fancy air filters. Well, some of us have fancy air filters in our homes. But wildfire or wildlife, they can't take shelter indoors. You know, they're exposed to this smoke year round. They're exposed to it 24 hours a day. And there's many dangerous things about smoke. Smoke contains a variety of sizes of particulate matter. We're all kind of familiar, or at least if you live in a wildfire-prone area, with the wind kind of bringing ash into your neighborhood and it might fall on your car. And while we can see the ash, there's many different types of particulate matter, and the most dangerous sizes are particulate matter 2.5 microns and smaller. Those are the ones that get stuck in the lungs and move on to the cardiovascular tissue. And overall, it can increase a variety of health impacts across different systems, including the lungs, the cardiovascular system, and the neurological system. There we go. So I've already talked a little bit about why smoke is bad, but I think there was a really interesting paper that came out in 2023 out of a university in Australia, and they used interesting complex modeling tool to show how the smoke persisted in the atmosphere and how it moved around the globe. And it persisted for up to 6 months in the atmosphere, which I think is just kind of insane to think about how it's not just persisting in the atmosphere, but it was moving around the globe as well. And smoke from the 2020 wildfires in Colorado and the West Coast went as far as Europe. And I think this also increases avian exposure because when we think about especially these neotropic migrants that are breeding in the boreal forest, you know, they're exposed to smoke in the breeding ground potentially while they're raising their young, and the chicks are exposed to smoke during the first important months of their life. They're exposed to smoke during their migration, especially in the Rocky and the Pacific Flyway. And then many of them are exposed to smoke in the Amazon River Basin as well, where there's some major wildfires. And I think research in this area is small, but it's like growing every day. And research so far is focused on kind of two impacts of smoke, and it has touched on the impact of behavior, on behavior, and the impact on physiology. This is a well-known publication amongst the bird wildfire smoke folks, and I think it demonstrates the impact on behavior really well because on the left-hand side we have a map of greater white-fronted geese that have been tagged in their breeding ground as they migrate south to their wintering ground. And they have different stopover locations that they stop to refuel and feed and prepare for the next leg of their migration. And then on the right side is the 2020 wildfire year, and it shows just how much their route has varied due to that smoke, including that one that ended up all the way in Idaho. And when you're looking at the right side or the left graph that has all the little circles that shows their stopover spot, they have more stopover spots in 2020, and especially this bird that ended up in southeastern Washington. You know, I wouldn't expect a greater white-fronted goose to want to hang out in like the sagebrush steppe of southeastern Washington, so I can't imagine that that was an ideal refueling spot for that bird. And this paper also estimated that the additional mileage done by these birds in 2020 was around 700 kilometers per bird, and that caused an estimated energy deficit of 2 days worth of forage. So these birds are in unideal habitat for refueling, and they're being pushed up to 700 kilometers off their migration path. We're not really sure how that's impacting their physiology, their survival, and things like that. So a lot of the research on pollution and physiology is done in industrial areas, which makes sense because we can choose an industrial area and a non-industrial area and have a nice control and experimental group. But I think this study on house wrens by Krieg that came out just this year is very interesting because she already had a plot of nest boxes set up and was able to continue her research during a smoke event and showed depressed nestling growth in the smoke season compared to the other seasons. And then for the industrial side, there's a lot of research on house sparrows and pigeons and some of these species that we're all familiar with in kind of industrial and urban areas, showing immune suppression and increased oxidative stress among birds with high pollution exposure. So as I kind of jump into oxidative status, if there's any chemists in the audience, I want you to bear with me while I simplify everything excessively. So we're going to think of oxidative status as a balance. And on one side, you have oxidative damage, and that's your reactive oxygen species. And on the other side, you have your antioxidant capacity. And there's two very general types of antioxidants, and those are your dietary, things you get from cranberries, carrots, those colorful fruits. And then there's endogenous antioxidants, and those are things that your body makes naturally, like glutathione, uric acid, and superoxide dismutase. So reactive oxygen species, which is the oxidative damage side of this balance, start in the mitochondria. So it starts in the electron transport chain, and these negatively charged molecules, often associated with oxygen, H2O2, hydrogen peroxides, they kind of escape the electron transport chain, and they can grab onto different parts of the cell. And that can be DNA, RNA, or in many cases, the phospholipid bilayer. A lot of birds, the composition of their phospholipid bilayer changes depending on the diet that they're eating. And during migration, specifically, birds are consuming a lot of polyunsaturated fats, which are a highly oxidizable fat. So during migration, they are potentially more at risk for oxidation than other times of the year when their diet might be more varied or have a larger variety of fats. So these reactive oxygen species, they kind of bounce around and they cause damage in the cell, and many times it can cause the death of the cell. And if it doesn't cause the death of the cell, it can cause damage kind of throughout the cellular bodies. And it can also kind of create a chain reaction of damage within the cell between different organelles. And here's a couple of publications showing some research that has been done on oxidative damage. So Bizet et al. In 2008 showed that alpine swifts with high year-to-year oxidative damage had reduced fecundity. Antioxidant capacity, so that's just like circulating antioxidants in plasma, were significantly lower in urban areas. And Scopoli's shearwater showed reduced fledgling success when they had chronic year-to-year high oxidative stress. So now we're going to flip-flop to the other side of the equation and talk about antioxidants. And like I said, there's two very general types of antioxidants. Our endogenous antioxidants, like I said, are uric acid, and that comes from the catabolism of protein, which is also very important for migration. Birds are burning a lot of calories, and when there isn't fat in their body, they're going to burn muscle. So birds that are emaciated might show a higher antioxidant capacity because they are burning that muscle. Glutathione is produced in the liver, and that's a very important antioxidant for birds and humans alike. And then superoxide dismutase is an intracellular antioxidant that's produced in every cell and kind of is supposed to account for reactive oxygen species as they're created. And then, of course, our dietary sources, vitamin A, anthocyanins, and vitamin E. And this diagram here is just kind of showing the very basics of what an antioxidant and a reactive oxygen species are. So the green antioxidant donates an electron to the free radical, so it satiates that negatively charged molecule. And depending on the antioxidant, it then also becomes a reactive oxygen species, hence the chain reaction that can happen depending on the antioxidant. But this is a frustratingly complex system for someone who's a field biologist. So a lot of these antioxidants can kind of recycle themselves. So they are antioxidants and then oxidative species and then antioxidants. It's a very interesting system. And here's some interesting research on the antioxidant system. So, and this is an interesting paper out of China showing the different antioxidant capacity across different tissues. So, in the same way that I said that the fat composition can vary amongst tissues. That means that oxidative damage can also vary amongst tissues. So on the left side, we have lung and heart, and this is total antioxidant capacity. And we can see the red is the higher pollution area, so we have lower antioxidant capacity, circulating antioxidant capacity in the lung and heart. But glutathione, which is the intracellular one, was higher in the polluted areas. So maybe there's some way that birds are upregulating these antioxidants in certain tissues, but not others. So again, there's a lot of unknowns in this balance of oxidative damage. And then the stork paper is two separate or three separate areas of one polluted area, one kind of in between, and one unpolluted area. And the chicks in the polluted area showed a lower circulating antioxidant capacity. So back to this balance, we want kind of a flood of antioxidants because having high antioxidants is going to help reduce reactive oxygen species, especially during these metabolically taxing periods like migration when metabolism is high, the creation of ATP is high, and the electron transport chain is just trucking away. And then if we have low antioxidants, of course, we're going to see kind of an increase in reactive oxygen species. So my hypothesis is that oxidative status markers, ROS and antioxidant capacity, are influenced by exposure to wildfire smoke, and I also included body condition because of that relationship between uric acid and protein catabolism. So my predictions, as pollution increases, I think ROS will also increase. Heavy metals can act as reactive oxygen species. They can also create reactive oxygen species. So as this smoke is being inhaled and these particulate matters are going into the circulatory system, there's that potential they are creating additional reactive oxygen species. And as pollution increases, antioxidants will decrease for a couple reasons. In theory, because these antioxidants are quenching the reactive oxygen species, but also because these transition metals can also act as ROS in the body, and that means that they're grabbing onto the antioxidants and depleting the body of its natural antioxidants. And then as emaciation increases, which is our scale of body condition, catabolism causes the creation of uric acid, so our antioxidants in theory should also increase. All right, wow, cruising through this. So we do our fieldwork at Clear Spring Ranch in southern Colorado. We're about 30 minutes south of Colorado Springs. This is a really great place to do migration research because you look to the west and we have our Rocky Mountains, and you look to the east and we have kind of like this high desert that's a lot of shortgrass prairie and a lot of cholla, which is not great for these like birds that are desperately looking for sources of fuel. We band 5 hours a day, 6 days a week during migration seasons, April through May, August through October. And this is just a list of birds that I tested. So we catch a lot of birds and I sampled a lot of them. And then these were the ones that we narrowed down to do the test on based on the different migratory strategies. And then also how many we catch. It's a lot easier to catch a Wilson's warbler than it is a willow flycatcher. So a lot of this was just volume. I also sorted out individuals with signs of breeding condition to make sure that they were in the kind of metabolic state of migration. So our condition measurements, we have an air quality monitoring station installed at our field site, and this monitoring station records, I think, down to the minute air quality, and it measures 3 different levels of air quality, PM, 10, 2.5, and 1, and we are working with 2.5 for this project. species. Obviously, we're interested in understanding how individual species are adjusting to uh, their metabolic changes during migration. We controlled for season because fall is kind of known for its wildfire season and spring not so much. But also it might allow us to capture some of the nuance and dietary changes between spring and fall migration. A lot of birds rely heavily on fruit in the fall, and fruits are rich in polyunsaturated fats, and then because of that, they're rich in antioxidants. We collect a standardized fat scale, which is similar in theory across all of the banding stations in the United States and Canada. And then I created a Z-score with that to standardize across different species because different species have different migratory strategies and might collect fat differently. And we did the same for mussel. So mussel, we use this interesting device. We call it a mussel meter. We found, oh, I forgot to include the citation, but it's a really interesting paper. This mussel meter, it kind of scores emaciation. So when you're blowing on the chest of the bird, you can kind of see the keel, and these two plates slide across each other. This is just a 3D printed tool. And it measures the top of the keel and then the muscle tissue around it. So a bird that's more emaciated is going to show a higher number on the scale. So a high number would be like 4 millimeters. So it's still quite small, whereas a low number, a well-muscled bird, would be 1 millimeter or less. And again, I created a Z-score with this as well to help standardize across the species because, like a flicker is going to have a much, much larger scale because they're a much, much larger bird. All right. So I think one of the hard parts of studying wildfire smoke is the wildfire for a variety of reasons. Like we don't want to be out in the smoke either because if this is injuring the birds, it's certainly making us sick as well. And we don't really know when wildfire is going to happen. So in the history of Colorado, we have a lot of wildfire, so we felt like our chances were pretty good of having some days with severe air quality. And we got kind of lucky. I think the 5 or 6 days after the AOS meeting last year, we had some moderately poor air quality. Um, so we didn't really approach anything that, say, Los Angeles or anywhere in the Pacific Northwest or Australia is experiencing for kind of weeks on end. We had a very short window of moderate air quality. So it's hard to say, how severe the air quality has to be to produce measurable results, which is part of what we wanted to look at. So yeah, like I said, our air quality monitoring station monitors for 24 hours a day. So we just compared basically the bird the day of capture to the air quality the same day of capture. So in the field, we collected blood samples from all of our birds. Those blood samples were stored on ice until I could make it back to the lab the same day where I spun them in a centrifuge. And that's what this third picture is. The capillary tubes, the yellow on top is plasma, and the reddish part on bottom is the packed red blood cells. So I pull the plasma off the top and I put it in a little tube, and then we freeze that until we're ready to process it. And each of these individual samples is going to get 2 assays, or did get 2 assays, and that's to help me understand how each individual bird is managing oxidative stress. So the first assay I did was a total antioxidant capacity assay. So that is going to measure everything that's circulating in the blood, and that will include the vitamins like vitamin A and vitamin E, glutathione, uric acid. So it's basically just testing the plasma's ability to react with kind of these ROS that are in the assay. The other assay I did is, I call it the ODOG assay because 8-hydroxy deoxyguanosine is very hard to say over and over and over again. So I'm going to call it 8-ODOG. And this is a molecule that is created when DNA is oxidized. So it's a really interesting way to measure oxidative damage because it's measuring the actual damage to DNA. And that was an ELISA assay. And again, we did each of these on each bird so we could kind of get a snapshot of what each individual bird's oxidative status looked like. So here's my model setups. We are planning on running AIC on this, and I'm very early in the stages of running this and learning about AIC. So any feedback that folks have is very welcome. So my global model is on top. Our variables are particulate matter, season, my Z fat, and Z muscle, and then of course species. And then we made model combinations based on what we thought might be related or impact each other. So fat and muscle, there was no correlation between those two. Many birds that have kind of mild to severe emaciation can oftentimes have fat, and a lot of times birds that have a lot of fat could be in fine muscle condition. So we included those together without worry. Species and season are hopefully trying to capture some of those nuances in diet and, of course, migratory strategy. And then, of course, particulate matter. Oh, so each of these two models, antioxidant capacity and oxidative damage, I ran these models for each assay separately. So this is, again, very preliminary. So these are my top competing models. The intercept came up first, and then muscle and season. And these are all very closely weighted. So there's a lot of uncertainty in here, and I think going forward, we will have to do some model averaging to determine what areas or what model would even come out on top, and mess around with some of the fixed effects that we have as well. For 8 ODOG, for our oxidative damage, species came out on top, which I think isn't surprising because there was a lot of variety within the species. And I think that some of the other nuances might be lost within that because we have a number of species that we're testing. And PM did come up in my second model, but because the delta is 2, we have a penalty for that, so we did not include it as the top. So my very rough takeaways right now, as emaciation, oh, let me go back to this slide. So the muscle, Z muscle, so it had a positive association. So as muscle score increased, so as our muscle score became larger, which is what we wanted to see, our antioxidant capacity also increased, which I think matched our predictions if our models are stable, which we will find out in the next week or two. And dietary antioxidants are more abundant seasonally, so spring might be more metabolically, oh, and spring might be more metabolically active than fall. So there's kind of two issues with our season category in that dietary antioxidants are more common in the fall, and then spring is a shorter migratory period, so birds' metabolic activity is much, much higher in the spring because they're trying to get from, the tropics all the way up to the boreal in a very short amount of time, whereas in the fall it's kind of a little bit more relaxed. Oxidative damage varied the most within species, which I think makes sense. And the next steps is to investigate the species breakdown further. And then our low wildfire smoke, I think, limited our ability to measure the influence of PM 2.5 on oxidative status. And I think going forward, I would be interested in finding areas that can help me measure that without putting my own personal health at risk. There's a lot of really interesting current research in this. Again, I'm going to bring up the Krieg paper because I think these kind of long-term monitoring sites serve as a really great opportunity to study this because we already have data from areas and days that have low pollution, and then we're already ready to sort of deploy during these high wildfire smoke events. Really, ooh, I keep clicking it. Hazardous wildfire smoke alters the dawn soundscape, so I think it's really interesting how wildfire smoke might be impacting the behavior of just a variety of wildlife in a wild variety of situations. And then another paper from Knee-High, which I forgot to put the citation in, showed an impact of body condition and capture rates of birds in California. So I think there is like a small community of researchers working towards this, and I think there's a lot of potential in this area, and I'm excited to see what comes out of it in the future. And I think oxidative status is difficult to measure and understand. So again, I'd like to continue kind of pulling apart the relationships between all of these antioxidants and how they influence oxidative status. Most research is conducted on industrial pollution or in a lab setting, so I think getting out into the bird's natural habitat and looking at it in the context of their life histories is very important. And then again, it's difficult to mobilize and sample safely during wildfire events. All right. Thank you. There's so many people I want to thank. Obviously, my advisor, Dr. Claire Ramos, my committee members, Annette and Daryl, the Ramos Lab undergrads who have been the main force out at the banding station every day with me for all of my seasons. My partner Jordan, my lab mates Kelsey and Lacey, and then just like a whole host of friends who have been here to help give me feedback on many parts of this project. And then all of the funders who helped me fund this lab work, which was very expensive, and we're a very small school, so we're very grateful for the support from all of these various organizations who have helped me along the way. And that is the end. Thank you so much, everybody, for coming.
All right, great. Thank you so much, Megan. Great link from the molecular level all the way up to ecology here. We will start out with the question. We'll get to the questions.
Mhm.
So Olivia Sanderfoot asks, I'm curious if you explored how PM 2.5 impacted fat score. Did you run any models to look at how PM 2.5 affected body condition? So two questions there.
Yeah, so I think we did include a PM and 2.5 model in our both of our TAC and our oxidative status models, but it did not compete with the top models. And I th think it can be hard to measure that, especially in the project that I did, because it's hard to say where birds are coming from and where birds are going and how long they've been there. So I think maybe a more nuanced approach in terms of tagging a bird and seeing how long it stays and what the fat accumulation is over a number of days. And maybe that would have to be kind of like a captive study, just because it's hard just kind of pinpoint where that bird is at any point in time during the time that we capture it. Hopefully that answered the question.
And I'm going to Jamie Cornelius. I have just unmuted you or allowed you to talk if you want to ask your question.
Why, thank you.
Yeah, I granted you permission to.
Uh-oh, look out.
Yeah.
Well, first of all, Megan, great job. That
Thank
was a
you.
really nice talk. And I have been doing some similar efforts, so I completely feel your pain about the smoke and how
Yeah.
you really can't control it. So it's just up
Yeah.
to what happens. I was curious if you've tried doing any kind of grouping of species by like dietary guild. I'm seeing some, I don't have as many species in the sample size that you have, but
Yeah.
I'm seeing some interesting patterns about
Mm.
diet, and I'm curious if you've looked at it in that way.
I think it's something that I kind of talked about with my advisor, but I was concerned because there's so much variation between species, and a lot of our species will be like heavy insect eaters in the spring and then fruit eaters in the fall. And because I
Mm.
didn't quantify that in any way, I wasn't really sure if it would show up in the statistics in a way that made sense. So I decided not to include it for now. But I think maybe with a little bit more lit review on like maybe similar kind of um, inclusions, I would be interested in considering it.
Yeah, it's totally true that even really common species, when you go to look for actual real data on diet, it's can
Hmm.
be hard to find.
Yes.
Um, but I wonder if you just limit it to the fall when they're most likely to be exposed, or
Yeah.
just when you had those light smoke events.
Hmm.
Anyway, just something to think about because
Yeah.
I think we're seeing a little bit of signal there.
That's super interesting. Well, maybe I'll reach out to you because I'd love to hear more about it.
Sure.
And I'll
Yeah, I'd
chat
be
with my
happy to,
advisor and see what she says as well.
yeah, happy to chat some more about the various things we've been trying.
Cool. Sounds good.
Yeah, so much data. Great to hear how it could be partitioned to maybe
Mhm.
find some type of pattern like that.
Yeah.
Um, So, Tyne Baker asks, do you think it would be worth sampling on safe days at breeding or overwintering sites compared to the longer term measures of PM 2.5, for example, week, month, season?
Yeah, will you say the question again?
Oh, yeah, sorry. Do you think it would be worth sampling at safe days at breeding or overwintering sites and comparing to longer term measures of PM 2.5, for example, week, month, and season? I hope.
Yeah, I think it could I think you could, but I think I don't know because birds have completely different kind of metabolic states during their breeding and their migration and their wintering. I'm not totally sure how you would be able to compare like what was happening during breeding and then compare it to say migration or wintering, just because, you know, breeding is a very metabolically active period as well because they're feeding fledglings and they're singing and they're chasing other males and things like that, which is still very different from the period of molting and migration and then wintering. So I'm not exactly sure what the comparison would look like between those different states of metabolic status, I guess. I think that was the question.
I think so too. Safe
Okay.
days, does that uh Tyne, if we didn't answer your question, just
Mm.
drop us a message. Um okay, Mike Spalding asks, does the effect of smoke show up so fast? You were looking at the PM 2.5 count at the time of capture. Yeah, great question. Wouldn't it take a while for an effect to show up?
Yeah, I think that's a great question. And I did look into that, especially in terms of handling time, because I wanted to know, obviously handling can be very stressful for a bird. So I wanted to know what the impact was. And there is a little bit of research showing that oxidative status wasn't associated with handling time. And we also just ran a basic linear regression showing that our handling time did not impact the oxidative status. But that also shows that, it's not so immediate that it happens within a period of 15 to 20 minutes. And again, I think that's one of the things that is difficult about my project is we don't know how long these birds have been exposed because we're looking at such localized wildfire smoke. Maybe a bird came from the Pacific Northwest and was traveling through severe smoke the entire period it traveled and it arrived, and say another Wilson's warbler is traveling from, just north of Denver and there's no smoke and they might arrive here at the same time. We don't know where they've been. So it can be really hard to pick apart that a relationship between bird that might have been exposed for its entire duration of migration versus the one that was only exposed during what during the period that it's at our site now. And I think that would be something I'm interested in exploring is understanding how birds are migrating and if it's impacting their physiology, which again is very difficult to measure.
Challenging variables to consider here. Uh, okay, Emma Abel asks, I recently learned in my ornithology class about the avian respiratory system, specifically how delicate it is. Do you think this project can be expanded to look at the effects of smoke on bird respiratory systems?
Absolutely. I think a lot of the papers that I read to kind of prepare for this project focus, like I said, on industrial air pollution. So there's a fair bit of research on how pollution impacts um, the kind of like micro ecosystem of the lungs, how it impacts like white blood cells and some of these like really important immune response um, molecules that are present in the body. But I think there is still so much more to know about how wildfire specifically is impacting the lungs and the lung tissue. And I think especially how birds are surviving or not surviving. Like, what is the threshold of damage that has to occur before a bird can no longer survive?
Great. And sorry, Time Baker says, yes, it was a great it was a great answer. Safe days. Sorry, that was my fault for not knowing safe days just because I'm not familiar with the terminology
That's okay.
around smoke research. But safe days was a reference to sampling on days that are safe for the researcher's lungs.
Okay,
Um, and the motivation behind the question was I was trying to get at the idea of looking at smoke exposure over a longer period of time at a stationary location.
I see.
Um,
Yeah.
uh, Olivia, so I don't know if you have anything to add, Megan, but it sounds like your answer was right on point.
Perfect.
So Olivia Sanderfoot, I'm going to also allow you to talk, grant you permission, if you're
Hi,
ready to ask your question. We
can
can
you
hear
hear
you.
me?
Yep.
Oh, cool. I've never asked a question in a webinar before. I'm glad that that worked. Hi, Megan. Great talk.
Hi,
You are so good at explaining that background biology, just
thank you.
absolutely fantastic. So I know we've talked about your work before, and for
Mhm.
those who weren't at our AOS symposium, Megan and I, with Claire Ramos and Morgan Tingley, led a great symposium on all the research on wildfire smoke impacts on birds, and Megan's talk there was fantastic too. And I can't remember, did you band or study any resident birds? Because I think there's sort of this emerging question across this conversation this afternoon about how it's really difficult to think about acute and chronic exposure for resident versus migratory birds. And might be really interesting to think about how PM 2.5 at your banding site is impacting the resident birds because you just know that they're hanging out there. And I can't remember if you banded it,
Mhm.
but look at that.
Yeah, so I did collect a lot of blood samples from resident species as well, and they are currently resting very comfortably in our -80 freezer, ready for a excited graduate student to look at them. So,
Incredible. Yeah, well, I totally agree with all of the background and setup and motivation behind your hypothesis. Migration is just such a huge energy taxing moment for birds. I think it makes a lot of sense to study how wildfire smoke is impacting birds at this time and during this stage of their life history. But if you've got that data on resident birds, you
mhm,
could address some of these questions that are coming up about how PM 2.5 exposure during a longer period of time or really in that moment of banding might be impacting them. So awesome.
yeah,
Great talk. I can't wait to follow your research and learn more about what you find out and discover in this really exciting area of research. Congrats.
thank you.
All right, so any graduate students or future graduate students out there listening, the Ramos Lab has some resident bird samples to analyze for you, maybe. Um, so I just wanted to ask real quickly, I'm just checking all the different areas, but I just wanted to ask, so you use the TAC, the circulating antioxidant, I
Hmm.
believe, measurement
Yeah.
in the ELISA.
Hmm.
But in your background, were those the methods also used in some of those background studies used, or is there another method that you could apply out there, like that Swift and shearwater slide?
Hmm. I think the answer to that is there are so many methods, and I
Okay.
think that trying to decide which one I wanted to choose, I think caused me a lot more stress than probably it needed to, just because there's so many different antioxidants and they all do slightly different things and they
Mm,
all interact with different tissues and different organs. So it was kind of difficult to decide what I wanted to look at and if it made sense to collect plasma, because obviously plasma is the easiest to collect on a live bird. A lot of these tissues samples and a lot of these papers that do tissue samples, the bird is euthanized, right, to collect those tissues. And to collect a large sample size, you don't want to be euthanizing this many birds. So I think in the end, total antioxidant capacity through plasma was the most effective way to collect that. And especially in our case where we were working with migratory birds, I think it's different when we're looking at like pigeons and house sparrows, and people are like less
mm,
emotionally attached to those for better or for worse.
Uh, and I think you addressed this in your talk, but I just wanted to clarify in case I missed it. Um, is the idea that following pollution, they're using up their antioxidants that they have, or that they haven't made the same number of antioxidants total because they are somehow less fit in some way because of that smoke exposure,
Yeah.
or both? Yeah.
It is, as far as I've read so far, that the antioxidants are being taken up or depleted. So, we're gonna think of it as kind of like, um, I don't know, you kind of have like a library of antioxidants that are being produced by the body and taken in through diet. And then there's different types of ROS kind of pulling from that library. So you're going to have your normal ROS produced during the electron transport chain. So that's just your normal everyday metabolic rate. And then you're going to have things like heavy metal exposure, which is going to come in through smoke or maybe water or food or whatever. And those are also going to grab onto the antioxidants and kind of pull them. So if you have a lot of exposure, you're going to pull more antioxidants away from their normal metabolic antioxidants, which is going to leave you kind of without a library to pull from of antioxidants. And then additionally, those pollution-related metals, they can also create additional reactive oxygen species that come from the metabolic pathway. So you might start with one reactive oxygen species and then end up with two because of how these metals interact with those molecules. So it kind of logarithmically scales in terms of how these antioxidants are used.
Okay, got you. So much more the depleting as opposed to
Yes.
differences prior to exposure
Mhm.
between individuals. Uh, yeah, and then one more thing about the shear waters. So is the so I'm assuming that antioxidant study though is not dealing with fire unless the smoke is blowing over the ocean. Yeah.
Yeah, so that project is really interesting. So the author of that paper, Constantini, he's like the oxidative status guru. Like I'm gonna have like 25 papers for my thesis cited for him because he writes so many papers about this. But he monitors a colony of Scopoli shearwaters and samples them regularly year after year. So he was able to see birds that had this chronic oxidative status to see how it impacted their fecundity, their nestling success, and their chick survival, which I think is a really interesting project.
And so like what are some sources for that if not the smoke then?
Yeah, so I think that one he was just kind of looking at individuals with ox variation in high oxidative status. So naturally, different people, different birds, different animals are going to have different metabolic rates and different production of ROS. And if your body isn't producing as much
Mm.
antioxidants, you're going to have more reactive oxygen species. So that one wasn't necessarily showing an impact of pollution, just sort of that individuals with different reactive oxygen species or different statuses are going to have different, it's going to impact their life cycles kind of differently. So if you're adding pollution or migratory stress on top of that high oxidative damage that that individual already has, is when you're going to start to see maybe other things. So there's already kind of a variety between individuals. Did that kind of answer your question?
Yeah, that was great. Now I feel like I've read the paper. Yeah, that's fantastic.
Perfect.
Okay. Um, so just looking around here, Chris Ann has dropped the post seminar evaluation form in the chat. If anyone wants to go ahead and fill that out. And then lot of really positive comments about your talk, Megan. I think I speak for everyone when I say we really enjoyed it.
Thank you.
And um, I believe I've covered everything because there were a lot of questions. I
Okay,
think I got them all.
good.
Um, but I just want to thank everybody for being here. Thank you so much, Megan. Oh, wait a minute. Kathleen, I'm going to allow you to talk. Oh, wait, no, she put her hand down. Okay, false alarm. Okay, where was I in the thank you? Thank you for everybody for being here. Thank you, Megan, so much for your talk. And we will see you all next week.
Great. Thank you so much.